Well, when everybody's back, first of all, I'm very happy to be in Sweden and that I was invited to speak about a topic that has my interest in several decades already. And we are going to have a look at the so-called variation-inducing genetic elements. I think a lot of people do not realize, actually, that there are two sorts of science. There's materialistic science, and there is creation science. And what we do here is we do not believe that everything came by itself, everything made itself, like materialistic science believes, but we believe that everything has been designed by an intelligence. And I think this is the main difference, and most people do not know that. So I think we should stress that more when we give talks. It is very important to realize it. Anyway, I became a creation scientist about 20 years ago, when I also became a Christian. I wanted to know how did God create. The things that I watched, that I observed in the lab over the 1990s were so clear that they were constructed, that they were designed. The information that we found in these systems, it all points towards a designer. Who is the designer? That was my question. Around 2003, I found the designer. I was working in Sydney in those days, and I had a God experience, you can say. So then I knew, God, you are real. And I became a Christian. And I wanted to know, how did God create? I was a biologist, a molecular biologist, so I knew a lot about biology. And I wanted to know, how did you create? How did you create all these different species? And I started to look into that. And I found out that we have mechanisms in our genome that used to be thought of as junk DNA. They are actually not. So let's talk about these sort of things today. A new old look at transposable and transposed elements, as they're called now. Sort of transposons in the past. If we look in the Bible, what does the Bible say about species and speciation? Ola already had some nice quotes from the Bible. I will only give you one. And God said, let the earth bring forth living creatures according to their kind. And the earth brought forth is actually what we read. So God created something, not the species that we see currently, but something different. He created something that was able to cover all the edges of the world, that were plastic, not static, but plastic. They can adapt, they can respond to changes. They're able to generate variation themselves, and they're able to speciate, that means bring forth new species. The thing is, biologists do not really know what species are. Biologists cannot properly define what species are. And if you look into the literature, you will find at least 20 different definitions for species. So species is not a very good biological concept, not at all. The other thing is, if you look into the literature, you will find that about 10 to 25% of the current species can interbreed and produce fertile offspring, let alone the species that interbreed and do not produce fertile offspring. Which means that these are not species at all, according to the interbreeding definition. So there are many, many less species than believed there are. This also means, of course, that created kinds are not species, and species are not the created kinds. Also, if we look into current literature, what do we find? Superfast evolution, rapid changes, rapid speciation, extraordinary recent and rapid radiation, superfast evolution, rapid, superfast, rapid, superfast. This is not Darwinian evolution that takes ages. This is something completely different what we are looking at. Variation and adaptation, the phenomena that biologists refer to as evolution, occur very fast and not slow. It's completely different than the long ages and the mutations that have to add up over time and can be selected. No, it is different. I realized that in the 90s, early 2000s, that there must be mechanisms in the genome. Mechanisms of variation, adaptation, and speciation. And in discussions with other biologists, I did a lot of discussions early 2000, I became aware of several mechanisms in the genome that makes these things possible. And I described them in around 2003 and 2004, I described them in articles, which I later translated into Dutch, Back to the Origins, translated, Terug naar de oorsprong. And I also have a version in English, which I published later, but this is in fact the original version. And we also see that currently more and more people are realizing, even evolutionary biologists, that there is a mechanism that is responding to the environment, for instance, that then induces novel adaptations. And this is one of the books that I read recently from Peter Ward. He's an evolutionary biologist, and he calls it Lamarck's revenge. Why? Because the environment has important things to do and to drive adaptations. So this is very Lamarckian. We will not talk a lot about Lamarck today, but I realize that what he is describing in his books has also to do with transposons and epigenetics. And this is what we are going to look into today. Look at it. We already talked about the canids, the created kind canid. Here we see a lot of variation in the dogs, a lot of variation. It is tremendous if you look at it, how big some dogs are, and how small others are. And nevertheless, they can produce fertile offspring. They will never do in real life, because the one is too small and the other one is too big. It will not fit, but in the lab you can produce fertile offspring. You can fertilize eggs and produce offspring. The thing is, in the early 2000s, a lot of dog genomes were sequenced, and what did they find? The dogs hardly differ at the protein sequence level. Mutations are rather rare. They could not even make a pedigree, a tree, from these data. Why is that? I thought, they're so different. Why is that? Because there are mechanisms in your genome that are not discovered by the sequencing techniques that they had in those days. What they did, they only sequenced the exons, meaning that they sequenced the protein coding parts only. So there must be something in the genome next to the protein coding part that is driving this variation. Look at this. This is more recent. We can now look into much deeper and much more details in the genome than for 20 years ago. And we can now find dog and wolf-like social abilities, how they are associated to genetics. And what we do find that they are associated to signs, lines, TEs, retrotransposons. So most of the people have never heard of these abbreviations and words, but what are they? They are transposable and transposed elements. They're transposons and their relatives. What are transposons? Or as they're called now, transposable and transposed elements, or abbreviated TEs even. They are mobile genetic elements which can rearrange and restructure genomes. Restructuring of genomes. Who discovered it? Barbara McClintock, already in the 1940s. And in those days, she tried to convey the message that genomes are unstable, that they adapt due to this sort of mechanisms in the genome. And she wasn't believed. In those days, most people still believed or thought that a genome was very stable over very long times and not as plastic as she described it. So she had a lot of trouble to speak about it and to publish about it. Nevertheless, in the 70s and 80s, biologists rediscovered or discovered that she was right. The major part even of the genomes are made of this sort of elements. And then the scientific community was, of course, forced to give her a Nobel Prize, and she also received it in 1983, after 40 years, more than 40 years, because she was right. As said, most people didn't believe in a plastic genome, that it was as plastic as reported by Barbara McClintock, and therefore these elements have long been regarded as selfish DNA, egoistic DNA, junk DNA, remnants of viruses. Our genome has been made up by viruses. This is the message that we hear nowadays. It was still conveyed, this message was still propagated in 2002 in the study books. I have an example here from the most frequently used molecular biology of the cell book, Albert. What does it say in 2002? DNA sequences that ensure their own faith in the genome but do not contribute to the survival of cells that contain them. This is completely wrong. Completely wrong. In those days it was already wrong. So I checked the most recent issue of this book and it's no longer there. And that is very good. They realized that it is just nonsense. So they removed it from the book. More recently, since about 15 years or so, the view is completely changed from parasitic and egoistic to more symbiotic. We are dealing with symbiotic sequences. And we know this for a long time because creation scientists wrote on it as early as the 1990s that we are dealing with altruistic elements in the genome. I remember that Todd Wood wrote about it early 2000, and I didn't know that Todd Wood wrote about it, so I reinvented the wheel and came to the same conclusion, that it must be elements in our genome, and I understood that they were causing variation in the genome. So they actively generate variation in the genome. They are the source of variation of the genome. So and more people start to realize that. So mechanisms of variation, speciation in the creation framework, creation science framework. Genetics and epigenetics are playing very important roles. I have listed some up here. It is not exhausted, but I try to do my best. Polyvalence. There could be more than one allele, more than one gene created in the original kinds. So that you have a built-in mechanism of more information that can be released in the offspring. But also karyotype modifications, which means that the chromosomes may differ in that they duplicate, that they fuse, that there are fissions or inversions and so on. And what does it mean? It means that it affects the gene expression. And what does that mean, affecting the gene expression? It means variation. So you can just, by reshuffling your chromosomes, you can add, without the addition of novel information, you can get novel contexts. And this is variation. Gene regulation, differential regulation of genes is variation. And you can do that just by rearranging or restructuring your genome. You don't need any information, new information for that. You don't need any new genes for that. Epigenetics is also recently become a very hot topic. What does it say? It is about the regulation of your genes, how the gene programs are switched on and off. DNA methylation is very important, but also histone modifications. And I think tomorrow we will hear a lot about this. I'm not going into deep today, but just to mention it, epigenetics is also a very important mechanism for variation induction. And interestingly, both are controlled and regulated and affected by transposable and transposed elements. So genetics, we all know what genetics is, the study of genes and how traits are passed down from one generation to the next. And epigenetics is a layer on top of that. It's added upon the genetics. It is the studies of the activity of genes, how they are switched on and off during developmental processes, for instance, in cell differentiation processes, for instance. On the other hand, epigenetics also refers to biological traits not embedded in the DNA that are acquired over the course of lifetime, but can be passed on from parents to children. This is also epigenetics. And we have to keep in mind that these two mechanisms, these two processes in our genome are important for the induction and of variation, genetics and epigenetics. So how does our own genome look like when we look at it in percentages? I already said we have a huge part of transposons, or transposable and transposed elements, as they are nowadays called. We have only a very minor part, about 2%, that is coding regions for proteins. A minor part, only the red part here. Then you have a regulatory part that we know of. It's in the introns and it is just upstream of the genes, which is called introns and regulatory sequence, about 25%. So we see that the coding part and the regulatory part that we knew of is about 25%, a bit more than 25%. This is already a lot. Further, we have unique non-coding DNA. We don't know what it means. It could be species specific. And we have a repetitive DNA, like simple sequence repeats. We didn't know until recently what they meant, what they mean, but they all seem to be functional in making up the three-dimensional structure of the genome, but also in regulatory control. That is not to say that every sequence in our genome has a function, but there is much more function in our genome than the only 20% that is associated with the protein coding genes. So most of the genome is really littered with these so-called TEs. And I made a classification here. If we look into the human genome, we have about 45% of transposable elements. It adds up to 3 million. 3 million. You can further classify them and split them up in so-called DNA transposons and retroelements that are RNA transposons. And what is the difference? The DNA transposons use a cut-paste mechanism to be mobile in the genome. And the retroelements, or the RNA transposons, use a copy-paste. So they copy themselves, and via an RNA intermediate, they can move to another place in the genome. That's the difference between those two elements. Most of the mobile elements in or most of the RNA transposons or derived genetic elements are of these types. We find two of them, the so-called non-long terminal repeats and the long terminal repeats, the LTRs. And the most well studied are the lines in the science on the left-hand side, and the most studied on the right-hand side with the long terminal repeat elements are the so-called ERVs, the endogenous retroviruses. And why are they called endogenous retroviruses? This is also an interesting point, I think, because they look like retroviruses, like a certain type of retroviruses. And this is why they think that all these elements, the lines, the signs, etc., and the ERVs, come from integration, ancient integrations of these retroviruses. I think that is completely mistaken. I think it's the other way around, that some retroviruses can origin in the genome from these elements. And how would that go? Well, we know we have this HERV-K transposon in our genomes. And it only has to take up a very small part of a gene, which is the SRC gene for here, and this is a gene which is very important in regulatory processes of cell divisions. It's one of the so-called proto-oncogenes. And what did it do? It took up an on switch of this gene, integrated it in the sequence of this ERV-K, and now it became an RNA virus. This is one of the most simple RNA viruses that we know of. So you can argue that from these genetic elements that are present in our genome, every now and then a virus emerges. And this is one that is also found in real biology, and it's an oncovirus. So it is causing cancer. And why is it causing cancer? Because it took up the on switch only of a gene that is normally controlling the cell proliferation. So it cannot be switched off again. So in just a single step, you can create from an endogenous variation-inducing genetic element, you can generate a virus. So you start also to understand where some disease-causing viruses have their origin. In the genome. It happens every now and then in a fallen world. Anyway, as said, genomes are really littered with these TEs. I already mentioned people, humans, have about 45%. But other organisms sometimes even have more, like frogs. Other organisms have less, like for instance baker's yeast. But nevertheless, they have these type of things. So all organisms that we have studied so far, including bacteria, have TEs, transposable and transposed elements, DNA elements or RNA-mediated elements. Here we see a very simple overview. As said, bacteria have so-called IS elements, insertion sequences. What do? They generate variation in the genome. Also yeast, insects, mammals, they have this sort of elements in their genome. What do? They can generate variation in the genomes by affecting genes, by affecting gene expression. This is what they do. In humans, we see signs, we see lines, and we see ERVs. Here, we all have them. These are very complicated elements. As said, they use an RNA intermediate to translocate, leaving a copy of themselves. So in fact what they do, they generate variation, but the position where they were, which is also driving your variation and your gene regulation, is maintained. So you drive variation, but the old variation is maintained. So genomes are really full of them, and all genomes contain them. Nowadays, we call them transposable and transposed elements. I will make a brief excursion into the ENCODE project. This is the functional studies of the epigenome. Why will I do that? Because they analyzed the genome as a language. They wanted to know what elements are there in our genome and what do they mean. There are not only protein coding genes, there are many more genes, there are RNA genes, and there are all these elements that we don't know what they do. And they started out to look into it specifically, and they want to map all DNA elements and all their functions, and all the mapping of the epigenome. So this is what they do. What did they found so far? As early as 2007, if I'm not mistaken, they found that 93% of the genome is actively transcribed into RNA. It's highly active. In 2012, they reported that 80% or over 80% of the genome has a biochemical function, which is very, very contested. And why? Because an 80% functional genome couldn't have evolved according to the evolutionists. Why that is so, you can read in the literature, or you can ask me later. I'm not going into that. But almost fully genome cannot have evolved. So there's a lot of contention about these data. The ENCODE projects themselves, they never denied that it wasn't so. They still say. They don't say it anymore, that it is 80%, but if you add up all the functions that they find in the genome, it must be about 80%. Let's see what they found in 2020. They find about 20,000 protein coding genes. They find about double the amount of non-coding RNA genes. They found more than 4 million regulatory sequences, genetic switches, 2 million open chromatin regions, which is also accessible for your transcription factors. 750,000 regions with modified histone proteins, regulatory stuff for opening up and closing your programs in your DNA. 1.2 million regions for DNA chromatin binding proteins, transcription factors, all switches, genetic switches, and more than 1 million, or about 1 million transposable and transposed elements. So this is the new genome that we know around 2020. And as you can see, if you add everything up, it's much more than 50% functional. So in a conclusion, you can say evolution cannot have happened. We are not the product of evolution. It's impossible. So we are going to have a look at these big part of the genome, transposable and transposed elements. In 2009, when I was doing my studies as well in the lab, I encountered this type of research because I was in regulatory genetics, how a gene switched on and off. And this just appeared after I wrote my book. And it said, it reads the regulated retrotransposon transcriptome of 1 million cells. Well, for most people it doesn't say anything, but what does it mean? It means that the retrotransposons, the lines and the ERVs, are actually regulating the expression of genes. It's not the promoters or the enhancers, as we used to think. They act as distant promoters or as distant enhancers. This was really a seminal paper because nobody had expected it. Transposons are genetic and epigenetic switches. They conclude that retrotransposon-driven transcription has a decisive influence on the transcriptional output of the mammalian genome. And you can understand it because if you think about it, we have many different cells, and all these cells have to be differentially expressed in their gene programs. We'll come to that later. Here is another seminal paper. They show that line activation after fertilization regulates global chromatin accessibility in the early mouse embryo. What does it mean? It is a factor, a very important factor, that opens up the programs and closes the programs that you need in the differentiation and development of an organism. We are dealing with switches, genetic switches. And now we are about 20 years later, 15 years later, and now it is a common knowledge that transposable elements are essential elements in the control of gene expression. They're essential. You cannot do without. The junk DNA of the past century is essential for gene expression. So, TEs are tissue-specific epigenetic regulators of gene expression and cellular responses in health and disease. This is what they concluded in 2009. TEs are mobile genetic elements that populate genomes where the host largely controls their activity. This really is still referring to a host genome as if we are dealing with virus invasions. No, it's just a part of our genome. This is a sort of brainwash talk that we are dealing with viruses. We are not dealing with viruses. We are dealing with a unique regulatory part of our genome, which we are discovering only now, because it was junk DNA. It's no longer regarded as junk DNA now. It cannot be regarded. Nevertheless, it is still regarded as remnants of viruses. Well, why? Because otherwise we don't have an explanation for that, where they come from in a materialistic view. We can understand them as genetic switches, as creation scientists. So, meeting report of 2018. The genome is very plastic. It's not static, as Darwinians used to believe and still believe. It's very plastic. Neuronal plasticity, learning processes. What is involved here? Also the TEs, transposable elements, in the brain. Every neuron, almost every neuron, has its own genetic makeup. How come? Because of these TEs. So it is a plastic part of our genome. And we still do not really know what it means, but the people who researched it, they think it is helping in learning processes, or even needed for learning processes. And they have good arguments for that. Also, they argue for organismal plasticity. What does that mean? Organisms are very plastic. It means that they can adapt, that they can respond to changes because of transposons. This was 2018. We creation scientists published this as early as 2002. I forgot Todd Wood, who published in 2002 about this topic. I published in 2009 on this topic. I said, okay, let's have a look at them, what they do. They must be variation-inducing genetic elements, 2009. Another one in 2009. And this is in fact what they are able to do. If we look carefully, and this is a sort of summary of my papers, they can rearrange the karyotype, restructure the chromosomes. What does that mean? That is nothing else than variation and speciation. They can also duplicate or translocate or parts of your genome. What does that mean? It changes the genetic output. It changes the gene expression. What is that? What is another word for that? Variation, adaptation. So everything, I realized then that everything that we see as evolution or as variation or as adaptation may be traced back to this type of elements. And this is in fact what I described in my book. So how do we see a gene nowadays? It's not a static thing, no. It is a coding part here. The exons is only the coding part. We knew already about the entrance. We knew about the promoter, which is driving this whole part, but you also need TEs to drive it and to regulate and control it. This is in fact what we discovered the last couple of decades, 2 decades. So the whole idea about a gene changed over the last 10 to 15 years. This is a gene now. You have protein coding parts, but it can also be RNA genes, and you have a whole bunch of TEs sitting in between. As said, you have millions of them. And this is what you get. You control gene expression at the genetic and epigenetic level. This is what they do. Today, a gene is understood as a coding island in an ocean of regulating and controlling genetic elements. Most TEs accumulate in this ocean, group themselves between the islands, thus changing the genetic and epigenetic regulatory context, hence affecting the output of the genes. We are dealing with variation-inducing genetic elements, differentiation, adaptation. This is what they do. I will give you a few examples now of a DNA transposon, what it is able to do, for instance. Here we have a very simple example. We have a pigment gene which provides a color. This color could be a protein. It is a protein. Then a transposon jumps in, it interrupts the gene and the color disappears. It can also be a different gene, but for color it's a nice example. Because we have seen this in mice, for instance. You have a so-called mutator transposon, and it was one of the observations of Barbara McClintock of the 1940s. It moves in the pigment gene, and what do you get? Beautiful variation. You don't need 10 genes for this beautiful variation. You need only one gene in one mobile element. And you can cause a lot of variation in your programs. The same holds for grapes. We know that there is a GRET1 transposon integrated in the white grapes. It's interrupting the gene. This is variation. You have a variation inducing genetic elements. One of the big surprises about 5 years ago or so, we know all about this so-called proof or evidence of evolution, the puppet moth. We know them. Nature, 2016, so almost 10 years ago. It is a transposon. They argue here that the discovery that the mutation itself is a transposon will prompt further debate about the importance of jumping genes, which are transposons, as a source of important phenotypic innovations. How does it work? We have a melanin gene, which we call Carbonaria locus, produces melanin. It produces melanin only in certain types of cells. It's regulated and controlled. Then we get the CARB-TE in, and all cells start to produce. So in fact, it is upsetting your production. This was one of the surprises. Another big surprise is that they are active in the brain. I already mentioned it. A big surprise. Almost all neurons are different at the DNA level. Why? Because of insertion of this line 1. It's a type of transposon. What does it do? I already told you. It is probably involved in learning and probably also in personality development. Very, very big surprise. But it is inducing variation in your brain, so that nobody is the same. So there are variation inducing genetic elements even in your neurons. So this brings me to the first take-home message. All eukaryotic genomes are full of transposons, the so-called endogenous retrovirus, ERVs and lines, and their remnants between brackets, and they're called signs and lines. They're really full of, all these genomes are full of them, and they are now known as transposable and transposed elements. They function as epigenetic switches in activating and silencing genetic programs, and they induce variation. And now we go a step further, we look a bit into the epigenome, epigenetics. I think this is one of the most visible, most nice examples of what epigenetics can do. They change a caterpillar into a butterfly, and both programs run in the same cells but are differently regulated. There are two distinct animals in the DNA of a butterfly egg. Two completely distinct, and it has to be simply regulated and expressed. It's a bit like Lego, you can say. With Lego you can build a lot. With the same genes you can build different organisms, only by regulating them differently. And this is also what we have in our body, but in all multicellular life forms. Here we see differentiation. From one single cell, one fertilized cell, you get about 300 to 400 different types of body cells. And even more of your neurons, as we recently discovered. How does it work? How does the epigenetic work? Well, first of all, you have DNA methylation, which is able to close off your DNA just by hanging a methyl group on your DNA at certain positions that are well defined, so-called CPG islands. What happens? You have a methyl group. It cannot be transcribed anymore, the gene. It's switched off. So you can control whole programs with it, DNA methylation. The other thing, on the other hand, you have so-called nucleosome modifications. This is the nucleosome. It is a complex of 8 proteins, and it has a very little tail, which is attached to that. It's sticking out here. And this can be modified. For instance, methylated, it can be phosphorylated, it can be acetylated, and this is also a sort code that is opening up your programs or closing your programs, depending how it is methylated, phosphorylated, and acetylated. And this is a research field on itself nowadays, how it works. You can talk hours about it. I'm not going to do that. I will only focus on methylation. Why is that? Because methylation has direct effects on your programs. They're closed. How does it work? Like that. The DNA is methylated and the gene or the program is shut off. So this is epigenetics in a nutshell. It refers to biological characteristics that are acquired throughout life. Cell differentiation, for instance. And it has two sides. Cell differentiation in the first place. Cells have to differentiate in different types of body cells. They all have the same DNA. So certain programs have to be switched off and others have to be switched on. A kidney cell and a heart cell have completely different programs activated. So you understand, I think. Some must be switched on, others must be switched off. This can be done by methylation. And on the other hand, we have acquired heritable traits. This is acquired DNA methylation patterns can be passed on from parents to children. This was also a big surprise. We thought that the complete methylation pattern was stripped off from the DNA and they could not be inherited. But now, the last 10 to 15 years, we find that also these patterns can be inherited. So this is a cell differentiation of a pluripotent stem cell, and you get different types of blood cells. I show it because I used to work in hematopoiesis, how blood cells form. This is very similar to pluripotent cell that is differentiating into all these cells. I forgot to say it. These cells is called a pluripotent cell. But this is very similar to how we think about created kinds and differentiation of species. It doesn't need novel information. It doesn't need novel genes. It only needs novel gene expression. So how could this work, these transposons? Here we have an example of how they are working. You have closed DNA and open DNA. If this transposon is relocated, it will also stop some programs there. So this is leaving a copy behind. So the old position is remained, the old variation and regulatory control is remained, and it is inducing novel variation. Is the process reversible? We still don't know. We still don't know. We don't know how they are controlled. But I have seen in the literature, I have seen some very interesting things where they show that it could even be excised and removed again. So maybe we have also this type of regulatory patterns in our genome. We don't know. We haven't looked into it. Anyway, I will show you another example of these transposons at the epigenetic effect. There is also a mutator gene, transposon. When it jumps in front of this so-called color gene, this pigment gene, the RFB gene, you get more or less methylation of it, and you get different sorts of colors, from white to light blue. So you can variate your expression of your gene, your output of your gene. And you see, you get very nice, colorful patterns of it. These are transposons. Variation-inducing genetic elements. More effects in plants. Here we have the shepherd spurs, which you can see all over the place, I would say, also in Sweden. They say here, TE insertions at the flowering locus explains to 12.5% of the natural variation in flowering time. So if you go to the south, they have a different flowering time than when you go to the north. How is it regulated? You don't need new genes for that. You only need new control elements for it. TEs are control elements. They control this variation. So, TEs are epigenetic switches. They are not junk DNA. They are epigenetic switches. I've tried to summarize this. On the one hand side, we see direct genomic effects. These changes do affect the DNA sequence, in fact, where they operate in. The effects are direct. On the other hand, we have epigenomic effects. The integration is not affecting direct in genes, but for instance, downstream methylation patterns. So although it is integrated in the DNA, it does not affect directly at that level, but at the epigenetic level. So TE or TEs modify the epigenetic genome and affect phenotypes by creating and or rewiring new epigenetic gene regulatory networks. This is nothing else than new variation, new traits that can be released. So it's all in the genome. Everything that we observed ever, in my opinion, can be traced back to variation-inducing genetic elements, except for maybe a few exceptions where point mutations and interruption of genes are also playing a role. But I think what we see here is a main part of the genome that controls variation and speciation. This brings me to the second point of functions that TEs have is to promote major structural chromosome changes, like duplications, like fusions and fissions of chromosomes, and also inversions. We used to think that this type of major structural changes just happen. But in biology, nothing just happens. If in biology things just happen, most likely you will have a disease afterwards. So these things are also mediated by repetitive units, repetitive elements, like these vikes that I showed you. And why is that? Because they can line up, as shown here. These are R1, R2, R3, etc. are the repetitive elements. And if they make crossovers, when they align the chromosomes and they cross over at the same place, there's no change at all. This is simply exchanged for that one. So crossing over the same genetic element, and we have millions, remember we have millions of them in our genome. If they cross over at the same genetic elements, there will be no change, but this hardly ever happens. They will not align perfectly, so you can expect duplications in your genome and deletions in your genome. So also this is mechanistically, it not just happens. You can even explain real big differences between populations in gene losses and duplications, as you can see here. If you have crossovers here, and you have 100 in between of these repetitive units, you will delete an entire part. And you can say, well, is this really biology? Yes, this is really biology. If you look into different populations of organisms, of what we call the same species, they can vary by 10% in their genetic content. Also people, as we'll see later. And even some of these TEs have their own recombination enzymes to promote these type of things. So it is all regulated and controlled. Nothing is left to chance. Do we have examples for this? I took one example here, duplication with a huge phenotypic effect. The dachshund or tackle got short legs. And we know in breeding, from breeding, we know that independently different types of dogs got short legs. And they are because there is an extra copy of the FGF4 gene, which was created by retro-transposition of chromosome 18. So it was created by a retro-transposon. Well, that's not of importance. I think this is also an interesting example. because we have some super genes that can be switched on back in the genome. And maybe you know something about these birds, because I think they are here in Scandinavia. They're also in the Netherlands. They used to be in the Netherlands. The ruffs, and what do the ruffs look like? The males have these beautiful colors, and they have also a beautiful color, as you can see, and the females don't. Now, is there one man and another man, and man 3? It is lacking all these male characteristics because there was a transposon-driven inversion. So you see this a lot, this type of variation. It is built in, and it can be unleashed in one single inversion. It doesn't take millions of years to evolve. It is simply switched in one generation, switched back, and you have it. So, similar things are going on in humans. If you take 2 people worldwide from different populations, they can differ up to 12% in their genetic content. That is tremendous. Even a man and a woman that sit here next to each other can differ by at least 3 to 4% in the genetic content. Nobody was expecting that. But variation is huge. And it has a lot of implications for how we think about what biology is, how it is functioning. And this also brings me in my papers to redundancy. We have a lot of redundancy of genes that are not really necessary, that you can lose. So you can create a lot of variation. So this guy says, who was one of the researchers, he says, each one of us has a unique pattern of gains and losses of complete sections of DNA. And he already wrote it, I think, about 10 years ago, or 15 years ago. And in the meantime, this has been confirmed. So there's a lot of indel mutations, huge mutations, of losses and gains in our genome. Okay, speciation, changing of the chromosomes, I already said it. Restructuring and remodeling of the chromosomes underlies macroevolution. It was already one of the postulates of Richard Goldsmith in the 1940s. And there's an obvious correlation between the increase in the number of chromosomes and speciation within the canine group. This is what Neil Todd said in his, I think in his 1970 paper. Yeah, and it's really worthwhile reading, because he looked into the chromosomes of the different sorts of canines, and he discovered that the chromosome, the amount of chromosomes is really predictive for speciation events. Here I have a few. I looked up a few. Maned wolf, for instance, they have a fused chromosome compared to dogs and wolves, which is 78. So, and if you look at it is here still in German, but if you look at the desert fox, you see that they have more fusions. So fusions, inversions, this sort of huge genetic variation induced by transposons lead to a lot of speciation events. Why is that? Because the chromosomes are unable to pair in meiosis. So they can even be reproductive isolation in this type of speciation events. So take-home message 2 is transposons function as genetic and epigenetic switches. Transposons promote structural changes to chromosomes, and together this promotes speciation processes. And also, more recently, this has been observed and integrated in the thinking of evolutionary scientists. And as well, I have here one from 2018. They say that the exceptionally high rates of speciation in the East African cichlids cannot be linked to an increased mutation rate. And if you know something about these cichlids in the African lakes, there are about 400 species, and they have been, the lakes were dried out in the ice age. So it must have gone very rapidly, even in the paradigm of the evolutionary thinking. Because they say these events took place in about 12,000 years. So in about 12,000 years, evolutionary time, you have about 400 to 500 species. And they say they cannot be linked to an increased mutation rate. Then they say in the genome of the cichlids, 3 waves of activities of transposable elements have been detected. So we again see here the transposable elements are driving the speciation events. It is built in. The insertion of transposable elements in the vicinity of genes has altered their expression. This is what they say. So this gene expression is in fact the driver, is driven by TEs and is the driver of speciation. Something similar was described recently for the radiation of the marsupials in Australia. Here we see that subsets of marsupials are defined by the absence and presence of groups of TEs. So you can simply define them on the level of this variation-inducing genetic elements. So you can argue these wallabies, they stem from one wallaby and they radiated because of the action of these variation-inducing genetic elements. And here, this is one of my final slides. There is a new evolution paradigm, you can say, that transposons promote speciation. And here is one that I discussed a couple of months ago with a group of creation scientists. Transposable elements activity is positively related to rate of speciation in mammals. So also here we see that evolutionary scientists are starting to realize that these transposons work or act like variation and speciation elements. So now we are coming together, the creation camp and the evolution camp. We have already seen that Ola presented this fixicity of species, which was wrong, and he showed the bushes of species, which is most likely how it is. And this one single tree is also wrong. So we can now start to understand how it works, and the evolutionary science also come in this direction. Well, this brings me to the conclusions. The genome of all organisms have built-in mechanisms to induce variation, genetic and epigenetic changes, which promotes adaptation and speciation. Speciation is associated with changes to karyotype and gene expression. There is no need for new information. Speciation events, number 3, are associated with bursts of TE activity. And this does not require eons of time. A burst means an explosion, an immediate activity. This is what they say now, also evolutionary scientists. Rapid radiation of created kinds to form species is a built-in characteristic of created kinds. It's built in. They don't have to wait for variation to be selected. They only have to, they will automatically speciate. And I think even selection is playing a minor role in this whole process. And then number 5, adaptation and speciation could be a Lamarckian phenomena induced by environmental cues. This is still speculative, but as we have seen, the integration of these TEs, these transposons in certain position, is also affecting your epigenetics immediately. So you can open up and switch off genetic programs very, very fast. As a response to environmental cues, for instance. And this is very Lamarckian, because it means that the environment is driving your adaptations. Well, and this has been forbidden for almost 200 years, these ideas. So now we're coming back to Lamarck. Am I the first one to argue in this way? No, there are many people who argued in this way, but apparently we have to argue always again, and we have to reinvent the wheels all the time because nobody's listening. The plausibility of life in the 1990s is also arguing that the genome can adapt itself, can change itself. Das Kooperative Gene, this is only in German. Goodbye to Darwinism, as a second title. Arrival of the Fittest, one of the professors in Zurich, Andreas Wagner, evolutionary biologist. He says that the genomes of bacteria adapt themselves because of insertion sequences. The paradigm shifters also argue in this way. Lee Spetner in the 1990s also argued in this way. This is more recent, I already discussed it. And my book also discussed it in 2009. So we are discussing the same things, but in different paradigms, as you can say. I think what we see in biology is clear indicative of creation. And therefore I will end with thanking the Lord, the designer, for science. Because science shows us the right way. Thank you very much for your attention.
Well, now we have 20 minutes for questions
and a coffee, maybe?
So, perhaps you can handle it.
Yeah, okay, Andy, you have a question.
I'm not a biologist, so forgive me if I'm venturing into areas that I express my ignorance, but I follow your logic that TEs are basically control mechanisms in the genome. Then you said that this leaves room right at the end, you were saying, so we need to relook at the mechanism. Now, there must be, therefore, some sort of edge to this possibilities, because we're not going to be back to Rudyard Kipling's, how did the giraffe get its neck?
It's not working like that. No, it's not working like that.
So there must therefore be a block on the phenotype changes, such you can't change the vertebrae in a giraffe's body.
No, you cannot do it like that.
Can you just comment on that? You have to repeat the question.
Yeah. Andy says, okay, he can follow the logic of these TEs generating variation, but then the leap towards Lamarck's ideas that the environment is driving the new characteristics which are then put in your genome, for instance. He cannot follow this logic. Is this a summary of what you said?
I'm saying there must be a limit.
There must be, yeah, of course there's a limit. The limit is set by the information of your genome. So a giraffe will never become a lion or something like that. This is easy to spot. How variable or how plastic is the neck of the giraffe? This is another issue. It can be sometimes 2 meters, and if you look between female and male, there's already a meter difference, about a meter. And if you look at the biggest males and the smallest males, it will also be about a meter. And this is what the epigenetics is. But this is not what we are talking about. We are talking about a different type of adaptation that is driven by the environment. There are now several very well documented examples, for instance, in mice that have been bred in a certain environment with a certain smell. And in this environment with this certain smell, they got also stressed. They got stressed. And now what they found is that if you breed these mice and you look in the second generation and you only give them this smell, they already get stressed. So a stress response is inherited through epigenetic signals. This is in fact what they show. You can also see it with some traits that have been very well studied in the Netherlands. In the Netherlands, we had a hunger winter in 1944. And what they found is that after 2 generations, you have this. Let's not make a shortcut. What they found in the children born after this hunger winter, they had obesitas, for instance, heart problems, and they could link it to the methylation pattern of an IGF-4 gene. It was not inhibited enough. And then they looked into the next generation, and they still found these methylation patterns on these genes. So the next generation also had these heart problems and also had these obesity gene expressed. So this is one of the things that is now very well documented, and this means that the stress in an environment is able to activate these transposons, which is then able to methylate your genes, which is transferred to the next and the next generation. And this is in fact Lamarckism. Yeah. So this is what in fact the newest findings show. So not that the how do we call it? The construction plan of an organism changes or can be changed because you need a lot of information for that. No. The expression of the information is able to change, and this can be inherited.
So could I just ask a subsidiary question? Very quickly, but what I'm anticipating is that this will be a new way of trying to promote an evolutionary explanation. Because people will be saying this is Lamarckianism is now valid and you don't need created original kinds.
The point is, yeah, I understand. I understand what you mean. The point is, well, Andy says this could trigger more evolutionary explaining, not in the sense of Darwin, but in the sense of Lamarck. The thing is, we have to define and look very properly into the genome. What are we talking about? Because we are doing science. You cannot just say, oh, this is evolution. No, what you have to do, you have to look into the different compartments. And what we are looking into now is not in the coding part of the compartment, because this is not changing. It's only the regulatory part that is changing. So the information to construct the whole plan of an organism is there. It's not changed. So I think I always try to discuss with people, with Darwinians, and they never define properly. This is the trick. So they will not define. But if you define it properly, you understand the genome, because it is a different compartment. We have several compartments in our genome. We have a coding part, an RNA coding. We have an RNA part. We have a non-coding part, and we have a regulatory part. So we have to really discriminate between that. Yeah, thank you for your questions. Oh, there are many questions now. Can you handle that? Yeah.
Okay. What time is it? Just 5 more minutes and we can have some coffee as well. Maybe.
Okay. So, first of all, what is the difference between transposable and transposed elements?
Okay.
You mentioned both,
Yeah.
but they have never been defined so far.
Exactly. Okay, transposable elements are the elements that are really transposable, mobile. But we have many more of them, also in our genomes, that are not mobile. They just sit there. They don't. They're in the human genome. There's one element, the line 1, which has the ability to move around. We do not know whether or not the others are able to move, but we have never seen them move. That's the point. So this is what they say. These are the transposed, because evolutionary biologists think that they moved in the past.
They have moved once.
Yeah, that's why they are transposed. Yeah, yeah.
Okay. So second question. As far as I know, transposable elements can be a source of double strand breaks. Is that right?
Yeah. Also, yeah.
This is a double strand break is the most dangerous damage to DNA. It causes cancer and other things. So 45% of our genome are composed of transposable elements.
Yeah.
Then this is highly fragile. It could be highly fragile if there was no mechanism to control it.
Yeah, it is controlled. Yeah.
To control it must be much more sophisticated and efficient.
Yeah.
To constantly prevent those
Yeah.
breaks
Yeah.
or to repair them.
Yeah, absolutely. You are right. I didn't go into that. Okay, he says that because our genome is 45% made of this sort of elements, it must be kept silent because they're involved in all sorts of double-strand breaks in our genome, which is derailing our genome, which can cause cancers, for instance. And then I said, okay, I didn't go into that, but these transposons and transposon-like elements are kept silent in our genome. They are methylated, for instance. There is a whole pathway that is involved in keeping them silent. As soon as they are becoming active, they are being silenced by the so-called PWE pathway. What is it doing? It is capturing these elements and putting them to rest, degrading them, because they are not. It is a controlled and regulated system in our genome. It is stringently controlled. The thing is, in my books, I talk also about the curse. What is the curse after the fall? What is aging? I think these two things can be explained by this regulatory part of our genome. As soon as the regulation over the control of this TE compartment of our genome is getting less and less because of mutations, it will add up, and I think after 50, 60 years, it is simply so damaged that it will upset the control of our genome. And this is the aging process, as far as I'm concerned. And this could be the curse.
Okay, I think we could have some coffee now and have some questions perhaps during the coffee break. And then we have other opportunities as well to communicate and ask questions and so on. Okay.
Yeah.
Good. Thank you very much for listening.
Thank you.